Now we're going to move into field studies for the last two speakers. So next we have Dr. Rintis Novianti. She's the head of the Malaria Pathogenesis Unit at Eichmann Institute of Molecular Biology. So she's here all the way from Jakarta. And she's going to be talking about the soldier relapse model that she has worked on with Dr. Baird and some of the amplicon deep sequencing we've done on some of these soldier cohorts.
Thank you, Jessica, for organizing this very interesting symposium, and also to Kevin Baird, who already always have a good source of person to discuss with. My name is Rintis Novianti. I'm from Eichmann Institute of Molecular Biology in Jakarta, and I'm also new in learning relapse. I learned so many relapse phenomenon from Kevin, from his soldier study done in Indonesia. So my talk title today is on multiplicity of infections in malaria relapse among Indonesian soldiers. So as you all know that Plasmodium vivax is a global health problem in malaria endemic region worldwide, including in Indonesia. And it has been known that almost half of the population in Indonesia, about like 130 million people living in at-risk populations in 2010. And roughly 5 to 15 million cases of clinical malaria are due to Plasmodium vivax infection. So we enjoy the luxury of studying relapse mechanisms through the study on malaria in soldiers that represents the unique study populations to understand the biology of relapse. In Indonesia, normally the troops are routinely deployed to the remote area that are normally high malaria area. They consist about 600 people for a duty for about 6 months. And they're normally originated from the densely populated and low malaria risk in Java, and then deployed to highly malaria area, like in Papua, on the eastern part of Indonesia. If you see that relapsing soldiers coming back from that area, they normally experience about 80% relapse. That's due to, at that time, there's no protocol applied for the soldiers to take Primaquine. That's why the relapse rate at that time is pretty high. So I joined the study at that time, led by Professor Inge Sutanto and Professor Kevin Baird, in testing the safety and efficacy of Primaquine against Plasmodium vivax relapse in Indonesian soldiers. As I pointed here, that normally soldiers are coming from the less densely populated area, no malaria area here, and being deployed in Papua, where malaria prevalence is pretty high, and then after 6 months they return to the home base for another deployment. And normally during that coming back to their home base, they experience relapse. So I just wanted to illustrate you about the samples that we got from these relapsing parasites. So through the screening of the soldiers that are returning from malaria area, about 1,000 blood films tested, and we enrolled about like 116 samples to be randomized through different drug treatment. And all of the treatment arms here normally has relapsed soldiers. And the highest is, of course, the one that are not having Primaquine in their treatment, and the rest are having much reduced relapse rate. And similarly, I would like to point it out here that after 6 months in Papua, in the malaria area, the soldiers return, and then we screen for malaria. And if they got malaria, they will be treated straight away. And after that, they're given like 2 weeks holiday off from the home base, and then they return, and we started to collect anything or anyone who are positive in this day 0 and monitored for the relapse for about 6 months to 1 year follow-up. So during this one, some soldiers experienced several relapse episodes. So similarly in other areas in Sragen, we also collected kind of soldiers from malaria endemic area and then did randomization as well. And then we end up with relapsed soldiers. And we had basically 2 areas in Java that we did our study in 2010 and 2013. And we ended up about 67 of soldiers that are having second relapse. And first relapse is about 139, make up to 206 soldiers. So it is very potential to capture about 67 relapse pairs in 139 individuals. So we are going to genotype this using deep sequencing methods that I'm going to describe later. So the aim of the study is to investigate the relapse pattern using deep sequencing techniques, the techniques that has been developed by a group in the University of North Carolina by Nicholas Hathaway et al. And also we would like to measure the multiplicity of infections of the relapsing parasites. So all the techniques that we performed was done at the Eichmann Institute in Jakarta. So we had an ion torrent machine that allow you to do deep sequencing. So the deep sequencing is based on the MSP1 42 kilodalton region, and it's been chosen because it is highly polymorphic, and based on the analysis of 250 haplotypes, it has shown that there is a spike polymorphisms in one area here. Sorry. How can I go? One area here that is giving you a very unique and no similarity between different areas. So we use this area that give you a lot of polymorphic areas that is really good or suitable for doing analysis of minority variants in relapsing parasites. So the haplotype was actually calculated based on more than 1% within host frequency, determined using the algorithms called SIGDEEP, firstly invented by Nicholas Hathaway, and the clustering algorithms that takes into account PCR error, chimeric detection, and sequencing error. So we would see all the different clones here, the variations, so labeled with different colors, and then sequence and count rates to find relative abundance of each relapsing parasites. So this is very, very new methods, and it's very sensitive to measure variations inside the relapsing parasites. And the result was we found that from the 127 relapse isolates in 94 individuals, basically, with the median about 4,000 read depths, we found 28 unique haplotypes with at least 2 SNP differences between haplotypes within samples. There were 9 singleton haplotypes, only seen in one sample, and 42 variable sites within 107 best pairs of sequence. So each of the color here represent different haplotypes. So we found quite a number of haplotypes in here. When we look at the multiplicity of infections, we found that like almost 60% of samples were monoclonal. And the average of MOI was about 1.6 clones per sample. range from 1 to 4. And it's very interesting that we compared with our field study from the same area in Papua by Pafa et al. In 2017. We also found in the community settings, Plasmodium 5X, there the rates of the MOI is also about 1.6. So it is similar with what we found here with the PV MSP1 deep sequencing. And there are variations in the number of clones that mostly they are monoclonals here from the relapse one, and then it decreased in number of variants. So when we look at the relapse pattern about comparing the patterns of consecutive relapse here, it is very clear that we can actually see the two dominant or different patterns dominated. So half of the dominant variant was detected in both relapse episodes. And sometimes they're monoclonal and sometimes they're polyclonal, like those that are having the same color here. From the first relapse and the second relapse, they show similar type of clones. And they encompass about like 52% relapse parasite that has the homologous relapse. And the second dominant type was actually the heterologous relapse that comprises about 33%, and they're actually not having similar type of clones here from the first and the second relapse. So there are two dominant type of relapse in these soldiers. Interestingly, using the SIG-DIP analysis, we were able to detect the minority variant expansions that cover about like 15%. And this is really interesting and very important because using the conventional or previous techniques, we were not able to detect the minority variant expansion. And it is clear here that in fact, there are from the first relapsers, the clones that are not present as a dominant one, they become the dominant clones in the second relapse, similar with the other types. So it's very, very interesting findings here that we can actually see that perhaps from the first time the minority variant were not expressed, but because of perhaps we thought that some immunity on that minor variant not present in the first relapse, they emerge in the second relapse. So for the complexity of infections, relapse are often monoclonal, despite evidence of multiple clones in the liver. So we found that about 61% relapse episodes were monoclonal, but only 24% of subjects harbored monoclonal infections if considering second relapse episodes. And the relapse timing, we saw that the median days to first relapse were about 30 days, and median days between first and second relapse were about like 47 days. There were no association between days between relapses and relapse pattern, and no association between individual variants and early or late relapse. When we look at the relapse decrease, the genetic complexity decrease over time, we saw there that we compared the 33 paired relapse episodes. We can see, in fact, that between the relapse 1 and relapse 2, it becomes decrease in terms of the number of clones as the relapse days increase. And when we counted all the relapse episodes from 1 and 27 isolates, we found similar pattern as well. So about 55% of the first relapse compared to 71% of second relapse are all monoclonals, and the multiplicity of infections is ranging about 1.6 to 1.8 versus 1.3 in early versus late response, late relapse consecutively. and the number of MOI by one clone is reduced over 333, so approximately 11 months. And we can think of that perhaps monoclonality as a rough proxy of hypnozoid burden, that perhaps also a measure of clone force of infection. So in summary, by deep sequencing findings, after a year of deployment, most soldiers harbored multiple hypnozoids clones, but these often relapsed in a clonal fashions. Most of our relapsing pattern here were 61% monoclonal. And this is very interesting because we thought initially that the relapsing parasites is always polyclonal, but these findings find either way. Individual variants did not seem to relapse after prescribed latency durations, and multiplicity of infection may serve as a rough proxy of hypnozoid burden or force of infections. We will have future study. Our plan is to capture primary infection to compare tachy and bradyzoids. Because the limitation in this study, we don't know whether the soldiers that got the first relapse were actually from the primary infections, because we did not do interview from the soldiers whether they were at the first deployment or consecutive deployment. And we will use also the multi-locus deep sequencing to determine genetic relatedness between the clones. And at the moment, we have the clinical trial tafenoquine studies in soldiers returning from the same area as well in Papua. And this is a collaboration between Eichmann Institute, EOCRU, University of Indonesia, Indonesian Army Force, and sponsored by GSK. And we combine different drug regimens, including DHP with placebo. And the soldiers were monitored for 1 year for the incidence of relapse. And we will employ MS genotyping, microsatellite genotyping, that we already had a techniques set up at the Eichmann Institute. And also we perform multi-locus deep sequencing and with whole genome sequencing. So at last, I would like to acknowledge this kind of work, especially to Dr. Kevin Baird and Jessica. Sorry, I would like to thank the help from Dr. Jessica Tellin and Professor Kevin Baird for a very enthusiasm for asking me to join to learn about relapse genotyping. Also, I would like to thank the Indonesian soldiers as the participants in this study. Also, I'm very grateful that I have a very good team work at the Eichmann Institute who have shown their hard work to do the deep sequencing in a very short period of time. Last but not least, I also thank to the Professor Inge Sutanto from the University of Indonesia and Dr. Ernie Nelwan for organizing such a very large clinical study, and the lab from the UNC Chapel Hill for the help in the analysis of the amplicon sequencing. Thank you very much.
Thank you, Dr. Novianti. Any questions? Can't tell if people are leaving or going to the microphone.
Could you just clarify, I'm not quite sure if I got this, so the soldiers treated with permaquine, what was the relapse frequency in the ones that had permaquine combination with other drugs?
From memory, the soldiers that were treated with primaquine, we had different concentration of primaquine from 15 and 30 milligrams. Right. Those that are receiving less dose, they relapse more than those that are having higher dose.
In the highest dose regimen, what is the relapse frequency?
Relapse frequency sometimes like about 2 to 3. No? Sorry, maybe Kevin knows better.
It's about 14%. in all the studies we've done, if we sum them up, 14% of soldiers will relapse despite high-dose, directly observed permaquin treatment. We published a study earlier this year on one of those battalions, and we found that 95% of those treatment failures were associated with significantly impaired CYP2D6 metabolism.
Okay. All right. Thank you.